Good afternoon to everyone. Our first speaker is Ernamma. Her topic is ultrastructure of plasma membrane and membrane asymmetry. Please share your screen, Ernamma.
Yeah. Hello.
Yeah, hello, Ernamma. Please start your seminar.
One second. Is it visible? Hello.
No, no, it's not visible. Actually, please present your seminar.
Okay. One second.
Please share your screen.
Yeah. Now is it visible?
Yes, please continue.
Okay. Good afternoon, all. My seminar topic is ultrastructure of plasma membrane and its asymmetry. This is a very well-known topic to know us, and I just recall that. First history. The term cell membrane was coined by Sinagali and Kramer in 1855, and the in 1895, Charles Aldrin suggested that the plasma membrane is composed of lipids, and in 1925, he got proposed that the plasma membrane is composed of phospholipid bilayer, and plasma membrane is encloses every type of cell, prokaryotic and eukaryotic cells. Actually, the plasma membrane is physically separate the cytoplasm from surrounding cellular environment, and the thickness of the plasma membrane is 8 to 10 nanometers. And plasma membrane is a dynamic and fluid structure and forms the external boundary of the cells, and it is also act as a selectively permeable membrane. Uh, because of it controls the entry and exit of the molecules or ions. That's why it is called selectively permeable. And there is structure of plasma membrane, and the parts of plasma membrane for extracellular fluids, bilayer, lipid bilayer, channel proteins, glycoproteins, glycolipids, cholesterols. These are the parts of plasma membrane. And the fluid mosaic model is a very acceptable model. And uh, in 1972, Singer and Nicholson proposed the fluid mosaic model, and this is very acceptable model. In this model, membrane are viewed as a quasi-fluid structure. That's why it is fluid mosaic model. And in this, the proteins are inserted into a lipid bilayer, and then the Singer and Nicholson also proposed that the membrane as a dynamic structure, and the plasma membrane is made up of your proteins and phospholipids, and they are also said proteins are alpha globular proteins are present in the plasma membrane, and according to Singer and Nicholson, proteins are two types: extrinsic and intrinsic. I just tell later the types of proteins. And the Singer and Nicholson also tells the ratio of proteins and lipids varies enormously depends upon the cell type. For example, human RBC cell contain 43% of lipids and 49% of proteins, whereas mouse liver cells are 54% of lipids contain and 46% of proteins are contained. That's why the ratio of protein and lipids are varies depends upon the cell type. And lipids and proteins are capable of flip-flop movement. Flip-flop movement is nothing but movement of phospholipids molecule between two layers. That is called flip-flop movement. And there's the components in plasma membrane. And so all the plasma membrane phospholipid. For example, this is the structure of phospholipid, and phospholipid bilayer particle. Yes, this is the structure, and the hydrophilic head and hydrophobic tail are present. And in this hydrophilic head is also called polar head group, and the hydrophobic head is also called non-polar group. And hydrophobic is a two fatty acid chains are present. One is saturated fatty acid, another one is unsaturated fatty acid. In the hydrophilic head, phosphate and glycerol is present. Depend upon the which phospholipid is present. Phospholipids also differ from every cell. And membrane components, proteins are present and lipids, different components, carbohydrates, proteins, lipids are also present in the plasma membrane, and highly proteins and lipids are present in the plasma membrane. That's why it is called mainly made up of proteins and phospholipids. And actually, phospholipid is amphipathic in nature. That's why it is two types: hydrophilic head group and hydrophilic tail is present. That's why it is called amphipathic in nature. And these phospholipid is two types of movement is present. That is transition movement and flip-flop movement. Transition movement is nothing but the movement of phospholipid molecule is same layer. The movement is occur in the same layer, that is called transition movement. Flip-flop movement is the movement of phospholipid molecule between the two layers. It is called flip-flop movement. And these are the phospholipid cholesterols are present, proteins, glycocalyx are also components in the plasma membrane. And these are the types of membrane lipids. For example, glycolipids, phospholipids, sphingolipids are also present. Depend upon the cell type. Which cell type is present, that is that phospholipids are present in the cell. And glycolipids, triglycerol and glycophospholipids, ethyl glycolipids, sphingospholipids, glycolipids, for example. These are the structures, and these are also structures of phospholipid structure. First one is and the sphingomyelin, second one. Glycolipids and sterols are the structures of phospholipids. Carbohydrates, next one of the component, carbohydrate. Carbohydrates are also found in plasma membrane. Most carbohydrates are part of glycoproteins, and glycoproteins play role in interactions between cells. For example, addition. For example, in the addition, the glycoproteins play the main role interaction between the cells. And the proteins are wedged between lipids, which are the molecules that could not pass through ion channels or gates. And proteins in the cell membranes are integral proteins, are also called intrinsic proteins and extrinsic proteins. In the intrinsic proteins are partially or completely embedded in the phospholipid bilayer. Intrinsic proteins are, and extrinsic proteins are these are present or they are attached to the phospholipid head. Extrinsic proteins are attached to the phospholipid head, and these are the loosely attached. And fat soluble proteins are passes through the extrinsic proteins, And water soluble substances are passed through the intrinsic proteins. And the role is cell signaling, cell recognition, enzyme activities are involved in the. These are the different types of proteins present in the plasma membrane: extrinsic proteins and monotropic proteins, multipass integral proteins, single pass integral proteins. Multipass integral proteins are also called beta proteins, beta globular proteins. These are the other which are not present. Uh, PC is nothing but phosphatidylcholine. Choline. PC is 50 to 77% of phosphatidylcholine is present in the plasma membrane, and 80 to 100% of sphingomyelin is present in the plasma membrane, and phosphatidylethylalanine are 87 to 100% is present, and phosphatidylserine is 94 to 100% of present in the plasma membrane. Um, Phospholipid asymmetry in the erythrocyte membrane. This figure is shown that, and the colors are indicate the asymmetric distribution of phospholipids in this picture. Thank you.
Okay, thank you, Ernamma. Now, our next speaker is Pavana Monica. Her topic is viral genetics. Please share your screen. Hello, Pavana Monica. Are you in the meeting?
Good afternoon, everyone. Hello.
Yes, Pavana Monica. Please continue.
Good afternoon, everyone. Let me introduce myself. My name is S. Pavana Monica. I am here to give a seminar about viral genetics. Viral genetics is the study of the mechanisms of heritable informations in viruses, including genome structure, replication, and genetic change. Disease-causing viruses that are particular of interest. However, genetics also use viruses to help understand fundamental mechanisms of molecular genetics. Introduction to virus. Viruses are typically described as obligatory intracellular parasites, a cellular infectious agent that require the presence of a host cell in order to multiply. Viruses that have been found to infect all types of cells includes humans, animals, plants, bacteria, etc. Viruses do not undergo binary fission. Coming to structure of virus, a virus contain a nucleic acid core, which is DNA or RNA, the capsid, which is a protein coat surrounding the viral particle. A viral envelope is some viruses have the additional surrounding lipid bilayer membrane, which is called viral envelope. A complete virus particle is known as a virion. These are some variations in viruses which having a diameter. Tobacco mosaic virus have 20 nanometers and glycoprotein have 70 to 19 nanometer in diameter. Bacteriophages have 50 nanometers. Viral genomes, viral nucleic acids, which are DNA, RNA, linear or circular, in which DNA may be double-stranded or single-stranded. In RNA, DNA may be double-stranded or single-stranded. In linear or circular, smallest viruses have only 4 genes, while large have several hundred of genes. These are some classes of animal viruses. They are double-stranded, single-stranded, DNA and RNA. Viral protein coat is called a capsid. Capsids are built from protein subunits called capsomeres. Capsids are crystalline shell. They contain 1 or 2 types of proteins which contain any copy protein. Surrounds the viral is a protein coat. Viruses membrane envelopes that help them to infect hosts. A limited number of cells that infect is known as a host range. Coming to classification of viruses, viruses do not contain a complete system for protein translation. Therefore, every virus is dependent on host translation machinery. The Baltimore system explains that virus classification. Using this classification system, messenger RNA, that is mRNA, is the sender in different pathways to mRNA from DNA or RNA genomes denotes the different classes. The Baltimore classification initially included 6 classes of viruses. However, 7th class can be added to accommodate gap in the genome of Hepanoviridae, hepatitis B virus. The 7 classes of Baltimore classification are as follows. These are 7 groups, and there may be positive or negative DNA, single-stranded or double-stranded mRNA, and then translated to proteins. In this Baltimore classification, group 1 includes double-stranded DNA, that is mRNA transcribed directly from the DNA template. Example, herpes virus. In group 2, single-stranded DNA, mode of mRNA production is DNA is converted to double-stranded from before RNA is transcribed. Example, canine parvovirus. In group 3, double-stranded RNA, mRNA is transcribed from the RNA genome. Rotavirus. Coming 4, single-stranded RNA, genome functions as a mRNA. Picornavirus. In 5th group, single-stranded RNA, that is negative, mRNA is transcribed from RNA genome. Rabies virus. Coming to 6th group, single-stranded RNA virus with reverse transcriptase. Reverse transcriptase makes DNA from RNA genome. DNA is incorporated into host genome. mRNA is transcribed from the incorporated DNA. HIV virus. Double-stranded DNA virus with reverse transcriptase. Viral genome is double-stranded DNA, but viral DNA is replicated through RNA. Intermediate the RNA may be served directly. Example, hepatitis B virus. Coming to plant viruses, Plant viruses are viruses that infect all plants. Like all other viruses, plant viruses are obligate intracellular parasites that do not have a molecular machinery to replicate without a host cell. Plant viruses can be pathogenic to higher plants. Plant viruses are rod-shaped with a protein discs forming a tube surrounding the virus genome. This is the example of a plant virus, that is tobacco mosaic virus, which is a rod-shaped, and also first discovered virus. Animal viruses. Animal viruses are the infect host human beings also. Some cause illness and others not. Viruses infect all cellular life, and although viruses can infect every animal, plant, fungus, and protist species, each has its own specific range of viruses that often infect only that species only. Example, Ebola virus, chikungunya virus, virus. Coming to bacteria, viruses that infect and destroy the bacteria. Example, phages that E. coli T4 bacteria phase, lambda phase. They can be referred to as a bacterial parasites, with each phase type depending on a single strain of bacteria to act as a host. These bacteria phases can affect host cell in replicative cycles. A life cycle of a typical virus, in which bacteria phase can be in host bacterial cell. And then phase DNA in host cell. Then biosynthesis of phase DNA replicates and phase proteins. Then maturation of a new phase particles are assembled, and the cell lysis are releasing the newly made phases. These are two replicative cycles, nothing but lytic cycle and lysogenic cycle. Coming to lytic cycle is a phase replicative cycle that accumulates in depth of the host cell. The lytic cell cycle produces new phases and lysis breaks open and host cell walls, releasing the progeny virus. A phase that reproduces only by the lytic cycle is called a virulent phase. Bacteria have a defense against phases, including restriction enzymes that recognize and cut the certain phase DNA. This is the life cycle of a lytic cycle, in which the phase DNA injects its DNA into bacterial chromosome and phase circularizes, and certain factors, if it is determined whether it enter lytic cycle or lysogenic cycle. If it is entered in lytic cycle, biosynthesis can take place, and then lysis and release can be takes place. Coming to lysogenic cycle replicates the phase genome without destroying the host. The viral DNA molecule is incorporated into the host cell chromosome. The integrated DNA is known as a prophase. Every time the host divides its copies of the phase DNA and passes the copies of daughter cells. An environmental signal can trigger the virus genome to exit bacterial chromosome and switch to the lytic mode. Phases that use both lytic and lysogenic cycles are called temperate phases. This is the lysogenic cycle, in which lysogenic cycle phase DNA integrated into the host bacterial cell to become a prophase. Bacteria reproduce copying prophase and transmitting it into the daughter cells. If its cell division produces population of bacteria infected with the prophase, otherwise daughter cell with the prophase can be produced, then again entered to the phase DNA circulation. Coming to RNA viruses, retroviruses. In RNA viruses, the enzyme used is reverse transcriptase. Copies of viral RNA into DNA host. Viral DNA can be integrated into host chromosome is a provirus. can be passed to other cells also. Host RNA polymerase now transcribed viral DNA into viral RNA molecule. Retrovirus. Retrovirus is nothing but a human immunodeficiency virus which causes AIDS, acquired immunodeficiency syndrome. Envelope with the glycoproteins for binding to specific WC. Capsid containing 2 RNA strands and 2 copies of reverse transcriptase. HIV infection. The HIV particle which enters the host cells, reverse transcriptase synthesizes the double-stranded DNA from viral RNA. Transcription produces more copies of viral RNA translated into viral proteins. Proteins and viral RNA self-assembled into virus particle and leaves the host cell. Some cancers, viruses appear to cause certain human cancers like hepatitis B virus linked to one retrovirus leukemia transforms cells into cancer cells after integration of viral DNA into host DNA. Most tumor viruses probably causing cancer mutant events. Emergence of new viral diseases. There are eight causes which cause viral diseases. First, in mutation of existing diseases. RNA viruses tend to have a high mutation rates because of replication of their nucleic acid, lacks of proofreading. Some mutations create new viral strains with sufficient genetic differences from earlier strains. This is the case in flu epidemics. Spread of existing viruses from one host species to another. Hantavirus from mice to people. The one spread from a small isolated population to a widespread of epidemic. Example AIDS. Thank you.
Thank you, Panu Monica. And now for our next speaker is Anand Kumar. His topic is General Morphology and Functions of Endoplasmic Reticulum. Please share your screen, Anand.
Slides are visible?
Yes,
Hello.
Anand, it's visible.
I guess good afternoon to all. And my similar topic is general morphology of endoplasmic reticulum. This was discovered in 1902 in Italian scientist Emilio Verratti and K.R. Porter in 1945. Showed through ultron micrograph the cells contain the lack of like network standards for this cytoplasm. George Emilio Verratti and Porter 1945 mention a key characteristics of endoplasmic reticulum and ribosomes. Am I audible?
Yes,
Hello.
Anand, you are audible. Please continue.
Okay. In 1966, G. Pallade generated the transmission of electron micrograph elucidating the endoplasmic reticulum. He studied with the granules. George 1974 future described the section of endoplasmic reticulum and the Golgi complex. These are the scientists who discovered the endoplasmic reticulum. Definition: endoplasmic reticulum is a complex network of tubular membranes exclusively present in cytoplasm and eukaryotic cell. And the endoplasmic types and this can be divided into two types: rough endoplasmic reticulum, smooth endoplasmic reticulum. About endoplasmic reticulum, make and package of proteins and lipids, much like as a simple line found in eukaryotic organisms, form an interconnected network of lanterned members enclosed cases tubes known as cistern. Largest organelle in eukaryotic cell. It provides separate chemical environment which allows the correct protein folding. Functions of endoplasmic reticulum: translocation of proteins across the ER membrane, endoplasmic membrane. reticulum membrane interaction of proteins into the plasma membrane, folding and modification of proteins in the lumen, synthesis of phospholipids and steroids on the cytoplasmic size of membrane, storage of calcium ion in the lumen, and they regulated are released into the systole, carbohydrates metabolism. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum, it is highly covered and tubular. It forms an interconnecting system of pipelines which from a network throughout the cytoplasm. It is involved in the production of steroid hormones in the adrenal cortex and endocrine glands. Lipid composition of membrane is different to the cell complement. Components is large abundance phosphodiesterone is a very low concentration of cholesterol. It is also very fluid and disordered due to the larger proportion of unsaturated fatty acids. It is more prominent in some cells than in others, depending on the cell function. Smooth endocytosis major classes lipids, but mainly PC enzymes are membrane facing the cytosol, where the substrates are present in sequence almost the calcium 2 plus from the cytosol, and it is achieved in Ca2 plus from a high concentration of calcium binding protein within the lumen. Specialized smooth declared to release and reuptake of Ca2 plus. Most widely used an example is the sarcoplasmic reticulum found in the muscle cells. Calcium ion release in reptake triggers myofibril contractions and relaxation of respectively in muscle. Smooth endoplasmic reticulum is a structure of smooth endoplasmic reticulum. Rough endoplasmic reticulum. Lattice sealed sac that is continuous with the nuclear membrane studied on its outer surface of ribosomes found throughout the cell, but the density is higher near the nucleus and Golgi apertures. It has abundant transclone pores. Ribosomes are free to attach the sites of synthesis proteins and transport them directly into the lumen. Outer in the ribosome can detect this presence of ribosomal membrane in the given through the endoplasmic reticulum. It is a name. Rough endoplasmic reticulum is a structure of rough endoplasmic reticulum. Function of rough endoplasmic reticulum. Protein folding assemble of subunit proteins disupplied for bond formation. This requires an oxidizing environment, whereas the cytosol provide a reducing environment, and oxidizing environment is found with lumen, which also contain this sulfide is isomerase and enzymes an enzyme which facilitates this disulfide bond formation. Glyco. It is audible. Hello.
Yes, Anand, please continue.
Glycolization, the initial stages occur in specific asparagine residues through the claticulum cycle. Degradation of misfolded proteins through the ubiquitin proteasome pathway. The study of this endoplasmic reticulum, the major contribution to study of endoplasmic reticulum came in with a protocol published by the Palladic outlining in method of isolated endoplasmic reticulum derived microsomes. Microsomes represent small athenic versions of endoplasmic reticulum still capable for protein transpilation, glycosylation, Ca2 plus uptake, the release of steroid with ribosomes, rough microsomes. Ribosomes are always found on outside of surface, so the interior of the microsomes is biochemically equivalent to the luminous space of And if mRNA encoding is secreted protein translated on free ribosomes, protein produced was larger than normal secreted proteins. However, when microsomes are added with the translated proteins, cleaved into size. How to study is done, molecular biology cell. Thank you.
Okay, thank you, Anand.